Welcome you again. The next point in the unit plant tissue culture is the artificial seed. So this artificial seed, which is also called as synthetic seed. So synthetic, that means the artificial. So this synthetic seeds is made up from the somatic embryo surrounded by the nutrient medium, which is protected by thin chemical membrane. So as you know, in naturally, if the embryo that is covered with the seed coat, but here for the preparation in the synthetic seeds, the embryo which are prepared by the tissue culture, this embryo that will be covered with the help of some chemicals or make the chemical membrane around this embryo is called as synthetic seed. So, what are the features of these artificial seeds or synthetic seeds? So, these artificial seeds they are comparatively smaller in size and to the natural seeds. जो natural seeds हम जो plants के रहते उससे comparatively हम very smaller size में synthetic seeds बना सकते. Then the synthetic seeds they only contain the somatic embryo. So the somatic embryo which protected by an envelope with the help of nutrient medium. So this nutrient medium at the time of germination, they provided the or they nourishing to this developing embryo. So ultimately this medium that will be helpful for the seed in case of this developing embryo. Then the seeds have minimum essential amount of the food. So their size of the seed is too small. The seeds are bound by a synthetic membrane made up by the sodium alginate or polyacrylamide gel. So, for these preparing the synthetic seeds, mostly the sodium alginate or the polyacrylamide gel is used. So, they will be make the membrane around this embryo. So this artificial seal we can store it up to the 4 years at room temperature. They remain as it is up to the 4 years. These synthetic seeds they will be helping uh, in transporting from one place to the another. They seeds they will be germinate uh, or the germination of the synthetic seeds will be similar like the natural seeds. So in recently in different in different plant species these synthetic preparation of the synthetic seeds will be used. So how the exactly these synthetic seeds will be prepared? Let's discuss. So here, first of all, the induced artificial embryo by the somatic embryogenesis. सोमेटिक एम्ब्रियोजेनेसिस के थ्रू यहाँ पे एम्ब्रियो प्रिपेयर किए जाते हैं. देन दिस सोमेटिक एम्ब्रियो they will be kept in the 2% sodium alginate, then drop the embryos in both the calcium salt, that is calcium and nitrate, that is CaNO3 twice solution for 30 minutes. So these they resulted into these artificial seeds coat around the or coat around embryo and sodium alginate. So this so this So, with the help of calcium nitrate and sodium alginate, the synthetic embryo that will be covered by these chemicals, and then after this they making the membrane. So this membrane again they will be filled or like the beads, these chemicals they are covered that seeds, and again these seeds that will be sealed with the help of beads through nylon mesh. So the calcium nitrate solution can be recycled, and beads are nothing but the artificial seeds. So this the method in that only just the embryo that will be prepared from the somatic embryogenesis. This embryo can be transferred into these solutions, sodium alginate and calcium nitrate. So these they can make the covering to this embryo, and they can look like the beads. Then in for getting the same size beads, just sew into the nylon mesh, so all the seeds that will be in same size. So this is the simple method for preparation of the synthetic seeds. Then there are the types of artificial seed. So the first type of this artificial seed is the encapsulated seeds. So encapsulated, that means the seeds they are individually enclosed in a capsule. Each capsule consists of an embryo, some amount of nutrient medium, and sodium alginate membrane. Encapsulated, that word meaning says that make the capsule of the embryo. Capsule banate हैं, those they are covered by the nutrient medium as well as sodium alginate membrane. Then a second type of the artificial seed, it is the seed gel suspension. In that, the embryos are placed in the semi-liquid or semi-solid gel, and result these seed gels they are suspension or seed gel suspension are formed there, and that will be like the seed gel suspension. It is the semi-solid gel. Then the third type, it is the seed type. In seed type, the embryo that are placed in between the two types of the semi-solid medium. So these medium that will be loaded here, that is sodium alginate or polyacrylamide gel. So like the seed arrangement, जैसे हम पॉड वगैरह लेंगे तो पॉड में कैसे एक के ऊपर दूसरा, दूसरे के ऊपर तीसरे से सीड अरेंजमेंट रहती है, तो उसी टाइप में हम यहाँ पे जो थर्ड टाइप है सीड टाइप, वहाँ पे हम एम्ब्रियोज है, एम्ब्रियोज प्लेस करेंगे, और वहाँ दिस में embryos again they will be covered with the sodium alginate or polyacrylamide gel. So this is the three different types according to the this arrangement of this embryo before the formation of the synthetic seeds. They are differentiated as encapsulated seeds. वहाँ पे जो भी एम्ब्रियो है, एम्ब्रियो हम कवर करेंगे न्यूट्रिएंट मीडियम एज वेल एज सोडियम अल्जिनेट से. देन सीड जेल सस्पेंशन में जो भी हम एम्ब्रियोज दैट आर केप्ट इन सेमी सॉलिड जेल्स. देन सीड टाइप में जो भी हमारे एम्ब्रियोज है, these embryos that will be arranged like the seeds, and then they will be covered with the nutrient medium as well as sodium alginate or polyacrylamide gels. Then commercial aspects of these artificial seeds are these commercially these seeds are very important because we can easily store it up to the years without loss of viability. Normally, when we keep the seeds in 2 or more years, then its viability will be और they will be not viable. वो अच्छे से उनमें germination नहीं होता. But here in case of the synthetic seeds, हम कितने भी years वो seeds हम that means वो embryo हम store कर सकते हैं. Then they are easy to handle because we make the small beads, so we can easily handle it. They can directly sown into the soil like the natural seeds. These synthetic seeds, we are making the make the membrane of polyacrylamide gel or sodium alginate or some nutrient medium. So we can directly use this seed, sown these seeds into the soil. Then there is no need of the hardening in greenhouse. We can directly use in field. So for small tissues, from small tissues, many embryos, that is many synthetic seeds can be obtained. So it is also the benefits of that plant tissue culture. Plants raised by synthetic seeds of some callus tissue are identical. So that will be there is no variations will be formed. जैसे हमने जो मदर प्लांट और जो एक्सप्लांट यूज किए थे एम्ब्रियो फॉर्मेशन के लिए उसका कन्वर्जन हम सिंथेटिक सीड्स में कर रहे हैं. तो जो भी हमारे जो न्यू प्लांट्स होंगे these all new plants they will be identical or same like the mother plant. So in that case there is a no variations will be observed in parental type. These plants they will be shows superiority like the parents and they will be the superiority will be maintained in the also in artificial seeds. So it is like the vegetative propagation method. In vegetative propagation, as you know, we can directly use the part of this plant and with the cuttings we can got the entire plant. Like same way it is जैसे हम जो एम्ब्रियो है एम्ब्रियो हम जो प्लांट सुपेरियर होंगे वही हेल्दी प्लांट होंगे उसका यदि हम यूज करेंगे तो अल्टीमेटली जो हम सीड सिंथेटिक सीड का यदि ग्रोइंग करेंगे तो वैसे ही प्लांट्स हमें वहां पे मिलेंगे. देन in artificial seeds, herbicides, pesticides can be added in nutrient medium. So there is no need to apply herbicides during sowing. It works very effectively. So जब भी हम सिंथेटिक सीड्स बना रहे हैं वहाँ न्यूट्रिएंट मीडियम में ही हम जो भी हमें हर्बिसाइड्स जो पेस्टिसाइड्स रहते कुछ यदि सीड्स के ऊपर कुछ पेस्ट अटैक करते हैं इंसेक्ट पेस्ट अटैक करते तो उसको कंट्रोल करने का काम हम विद द हेल्प ऑफ दिस एट द टाइम ऑफ प्रिपरेशन we can add some chemicals on that. Then and some extra nutrients may also provided in artificial nutrient medium. So no need to add the fertilization externally. Generally, the plants raised by artificial seeds are disease free. So the seed can transport it from one place to other. This nutrient medium of the artificial seeds, mycorrhizal or blue-green algae may be added to get plantlets. So natural seed production, germination, dormancy, etc. are the problems in some commercially important plants. But if we use these artificial seeds, then it will the answers or solution on that problem. So generally in some places the seeds goes into the dormancy, they will be non-viable. So if we make these artificial seeds, they will be helpful in some commercially important plant. So it is also the one of the important branch in plant tissue culture. They have used in many industries for overcoming such a problems regarding the seeds. So here these are the different commercial aspects of the synthetic seeds. Then I think last one it is the protoplast culture. So this protoplast culture, all of you got this point, synthetic seeds. So I can start the next point. All of you clear the synthetic seeds.
Yes, ma'am.
Okay, it is just a technique in that we can make the seeds or prepare the synthetic seeds with the help of somatic embryos, and in that the nutrient medium as well as the chemicals like sodium alginate or polyacrylamide gels are used. Then the next point in your syllabus, that is the protoplast culture. So a question asked like detailed account of the techniques of isolation of the protoplast from plant tissue. So here the first is the removing of the protoplast which are present in the plant cell. So protoplast is nothing but the plant cells without cell wall containing cytoplasmic mass bounded by the cell membrane. So here the cell wall that will be removed by the enzymatic process as well as mechanical process. So प्रोटोप्लास्ट मतलब क्या है कि जो भी सेल वॉल रहता है, सेल यदि आप सेल का स्ट्रक्चर देखोगे, सेल्स में प्लांट सेल यदि आप देखोगे तो उसमें plant cells without cell wall, which is called as protoplast. So, for removing of this cell wall, different process are used. That is enzymatic, some enzymatic as well as the mechanical process are used, and the cell wall will be removed, and then these protoplasts are used for the protoplast culture. So for removing of the protoplast from the plant tissue, there are two methods. First one is the mechanical process and enzymatic process. So let's see the one by one mechanical process of the isolations of the protoplast. In case of the mechanical process, uh, here the plant cells from the peel of the leaf will be collected, or first of all the peels of the leaf, peels that means the outer covering of the leaf. So वहाँ पे जो plant leaves के ऊपर जो outer papery और पेपर लाइक स्ट्रक्चर रहता है वो पहले निकाल दिया जाता है. देन दिस पील इज केप्ट इन हाइपरटोनिक solution. Hypertonic solution, that means if we kept it in hypertonic solutions, it occurs the exosmosis, and due to the plasmolysis, the shrinkage of protoplast takes place. And if the if we kept this pill in hypertonic solution, uh, they will in that cells the exosmosis will takes place, and due to this plasmolysis or process, the shrinkage of the protoplast that will be takes place, and these plasmolyte cells are operate under the microscope with microscopical or microscalpel to remove the cell wall. This isolation of protoplast, it is केरला। तो यहाँ पे सिंपली क्या किया जाता है, जो लीफ है, लीफ का जो आउटर कवरिंग रहता है वो रिमूव किया जाता है। रिमूव करके फिर वो जो पिल है वो हाइपरटोनिक सॉल्यूशन में रखी जाती है। जब भी हम हाइपरटोनिक सॉल्यूशन में वहाँ पे जो पिल है वो रखेंगे तो उसमें श्रिंकेज हो जाता है। जो आउटर लेयर है, श्रिंकेज होने की वजह से जो लेयरिंग है, लेयरिंग उसकी लूज हो जाती है। तो हम फोर्सेप की जो स्कालपेल की मदद से हमें वो आउटर लेयर वहाँ पे उसका रिमूव करना है। दैट इज द मैकेनिकल मेथड। सो दिस मेथड इट इज क्विक but it is less or less expensive. Its demerits are the normally only skillful person they can do this procedure. The new person they have not possible to easily remove the outer layer. So it is one of the demerits. Then sometimes these mechanical process they will be fail down. So it is also one of the demerit. Then isolation of protoplast cannot be stored or maintained. जब भी हम वो प्रोटोप्लास्ट रिमूव करेंगे डायरेक्टली हम उसे स्टोर नहीं कर सकते, तो वो नेक्स्ट उसकी प्रोसीजर करनी पड़ेगी। तो इट इज ऑल्सो द डिमेरिट इन दिस मैकेनिकल प्रोसेस। इन एंजाइमेटिक प्रोसेस we have discussed in next lecture. By using the different enzymes we can remove the cell wall of this plant cell and then they can be used for protoplast culture. So